Hi, my name is Paul van Dijk from The Hive. We have recently incorporated mass spectrometry proteomics data into cBioPortal. In this video we would like to give a further explanation of the data and how to access it. You've probably used cBioPortal to perform integrative analysis based on clinical, mutation and expression data, but a growing number of studies offer many more data types than you might think. This slide shows the different data types that are supported by cBioPortal. And recently we've started to curate proteomics data based on mass spectrometry for some studies as well. You can find all available studies on the landing page. But you might wonder, how can I get a good overview of what data types are available for each study? Now, here's the simple trick. At the top of the page, there's a dataset section. If you go there, you will find all studies loaded to cBioPortal. And at the right-hand side, you can see which data types are available. By default, you will only see these 4 columns, but you can select more to display. You can sort these columns in ascending or descending order, and this way you can quickly find the studies with the data types of interest. Okay, let's look at an example. The Cancer Cell Line Encyclopedia is a project developed by the Broad Institute in collaboration with Novartis, and it represents a collection of cell lines that serve as models for various cancer types. It contains over 1,000 cell lines which were used to generate a broad variety of genomic, pharmacologic and cell lineage data. Just recently a new dataset with intensive proteomics profile data for these cell lines was made available. And this work was prepared and reported by David Nusino and his co-workers. We will now replay some of their analysis in cBioPortal. The CCLE study can easily be found after entering CCLE in the search field. There are 2 versions available for CCLE, but only the Broad 2019 version contains the newly added data. We select the study and then click Query by Gene. And also make sure that you tick mark the z-scores of protein abundance. And if you would like to have some more information about this data type, you can hover over the question mark to get some more technical details. Then we select in Select Patient Case Set a subset of the samples, the ones with cell lines used in proteomics assays. And then we can enter the genes of interest. For example, ERBB2, and perhaps also EGFR. And then we click Submit Query. We will then be presented with the oncoprint, and from the legend you can see the color marks for highly and lowly expressed proteins. So if you then go to one of the columns representing one patient or sample and hover over, you can find more detailed information. In the publication by Nusino, they're primarily focusing on the correlation between RNA and protein expression. In the next example, we will redo this analysis in cBioPortal. We will go straight to the Plots tab, and on the horizontal axis we choose RNA expression. We choose the gene EGFR. And for the vertical axis, we change data type to protein level for that same gene. And let's put it on a log scale. And when you look at the plot, you see a straight line, and also the Spearman and Pearson correlation coefficients show a high correlation, which is statistically significant. Let's do the same for ERBB2. And here we see the same pattern and a pretty good correlation. Okay, let's look at another example. Can we identify proteins that are potentially associated with the disease by looking at microsatellite unstable cell lines? For this example, we will go back to the publication by Nusino and his colleagues. This publication has a supplementary table. It's called table S7. And in this table, which is actually an Excel sheet, there are the CCLE IDs for those samples which have an inferred microsatellite instability, the ones in orange, and those samples which have an inferred stable microsatellites, which are in green. So we're going to copy these CCLE IDs and move them to cBioPortal. So we will select CCLE 2019 again, and instead of choosing Query by Gene, we click Explore Selected Studies. So here we have the total number of samples, and now we will go to Custom Selection. We click on it, and we simply paste the CCLE IDs for the microsatellite instability group. We filtered to the listed samples, and you can see we are left with 99 samples. From this subset of samples, we create a new group. Let's call it MSI. Create this group. And then we will remove this filter again. So we will get back to the original number of samples. And now we quickly copy the CCD IDs for samples with a stable microsatellite, paste those samples IDs, filter to listed samples, and create another group. So we end up with slightly more than 1200 samples. And let's call this one MSS. So now we're having these two groups, we can start comparing them. So we select them both, click on compare, and we will now end up at the group comparison section of cBioPortal. And we will click on the protein tab. It will start rendering. And it will then show you a volcano plot, but also at the bottom a list of genes from which you can select one of interest. So let's say we're interested in ROD50, we will click on this one. And then a box plot is shown at the top. And here you can see that ROD50 has a lower abundance in microsatellite instability cell lines. Now you may wonder whether there are any other studies with proteomics data in cBioPortal. Well, in fact there are. Researchers at the New York University School of Medicine have integrated proteomics data from the Clinical Proteomic Tumor Analysis Consortium, in short CPTAC, and they integrated these with sample data from several TCGA pan-cancer atlas studies. If you take, for example, a look at the TCGA breast cancer study like is shown on this slide, you will find proteomic Z-scores from CPTAC in the genomic profiles for this indication. We hope you enjoyed this short demonstration on proteomics data in cBioPortal. If you would like to learn more, you can read our blog or subscribe to our YouTube channel. We are always seeking additional large-scale cancer proteomics datasets for inclusion into cBioPortal. So if you would like to donate data, please get in touch with us. You can find the contact details as well as the links to our blog and to our YouTube channels in the description below. Now, if you would like to learn more about other cBioPortal features and how-tos, you can visit our webinar section via the link provided in the descriptions below. There you will also find a link to our blog posts on the Hive website. So, thank you for watching this video, and also a big thank you to all the people that contributed to this great platform.